Okay, let's carry on talking about antibodies. So, heavy chain and light chain, we know about these, right? Each of them has, let's see, several isotypes. Yeah, so the heavy chain, there are 5 different isotypes for the M, D, G, E, and A. And here they're backwards. No, they're just in a weird, oh, they're alphabetical. Here's the M, here's the D, here's the G, here's the E, and here's the A. And yeah, each one gives the antibody a different function because it allows a different type of cell to bind to the antibody, or a different range of cells, because there'll be several cells that have receptors for each of these antibodies on them. Then your light chain is going to be one of 2 isotypes, either a kappa or lambda isotype, and is going to consist of a variable and a constant domain. And so these are when I say domain, I mean part of the chain, yeah, of the entire molecule. The other 2 terms that are useful to talk about antibodies are the avidity and the affinity. So affinity is how strongly it can attach at another single site. So how strongly the antibody can bind to the antigen, for example. And the avidity is the, yeah, the one is like given by a K on or K off rate, if you know about enzyme kinetics. So it's like how quick it is to jump on. That's what they're talking about in terms of the sum of the strength of binding. So I would say be aware of the 2 terms, and I would say we're not going to use them in great detail, because I don't think either of these is a perfect definition of what those terms can be. All right, so there are, 2 heavy chains and 2 light chains, each of which is made of several domains, and each of these immunoglobulin-like folds is like a domain. So here's our variable domain at the top, and here's our variable domain of the heavy chain and of the light chain. And then our constant region of the light chain is here, and our constant region of the heavy chain is this entire 3-domain piece. So domain is a term that you can use in different ways for a protein. It can mean a part of the protein that folds up and has its own structure, and is kind of like structurally independent of the rest of the molecule. And so if we're saying structural domains, we would say, yeah, 1, 2, 3 structural domains in the constant region of a heavy chain. The other term for domain, though, is a functional domain. And it refers to as many structural domains as you need to be able to do a particular job. And this particular job is binding to their immune cells. And so for that, we don't really bind to this part up here or this part. We only really bind to this part. And these parts are here just to hold on to the light chain and to hold on to each other, more or less. So you could say there's 1 functional domain, 3 structural domains, if that makes sense, on an antibody, heavy chain. The light chain is going to have either 2 structural domains, or you could say it's 1 functional domain, since together these have the function of binding to an antigen up on the top. And neither domain has this full function by itself. Okay, so immunoglobulin-like domain, we've already talked about these. These are the little things, the little domains that make up an immunoglobulin. They're mostly made of beta strands. If you remember back to our talk about proteins right at the beginning, that's those little ribbons where the protein's all stretched out. And yeah, they're little beta strands that are folded up into kind of a little barrel, so like a little circle of beta strands. And each one of those is an immunoglobulin-like domain. And so, yes, there are variable and constant domains that are part of the immunoglobulin-like domain. The variable ones are the ones that change in response to the antigen, and the constant ones are the ones that remain the same regardless of what variable domain is on there. All right, a beta sheet is what we were just talking about. So it's the type of domain that makes up an immunoglobulin-like fold. And so you can make beta sheets, just if you're curious, in 2 ways. They can go like left to right and then turn and then back right to left and then turn and then go left to right. So they're basically like zigzagging, but with the sort of flat parts in the middle lined up with each other and hydrogen bonded. That would be an anti-parallel beta sheet where the strands are going opposite directions. You can also have a case where the beta strands all point in the same direction, and the protein just kind of loops around behind so that each strand can be going in the same direction. That's fine. That's a parallel beta sheet. They don't happen quite as much, but there are some of these things. If I remember right, I think immunoglobulins, oh yeah, here it says, are anti-parallel, yeah, beta sheets. And those are the more common in all forms of protein. And the beta sheets are folded up into a beta barrel, which is a barrel shape, or the side of a barrel, if you like the side of a cylinder, made of little beta sheets. So yeah, that's what an immunoglobulin-like fold is. Each of the sheets is made of a bunch of little beta strands, and so the beta strands are the little parts that make up a beta sheet. And a beta sandwich is, any structure that's located, that has beta sheets on both sides of the molecule and something else in the middle, like an alpha helix. So there are beta sandwich structures also in antibodies. The whole fold is called an immunoglobulin fold, and so each of those little units, so there's going to be 4 of these immunoglobulin fold units on a heavy chain and 2 on a light chain. The whole thing is going to be an immunoglobulin-like domain. That's like the part that functions as an immunoglobulin. And then the immunoglobulin superfamily are going to be a large, large family. Oh my gosh, so many proteins that each contain one or more immunoglobulin-like domains. So an actual antibody is part of the immunoglobulin superfamily, but so is a bunch of stuff that's not really an antibody. Okay, here are some immunoglobulin-like domains. Look at them. So light chain constant domain, here we've got 7 little beta strands, and see how one goes up, one goes down, one goes up, one goes down, just like that. That is an anti-parallel beta sheet. And you can see that this yellow sheet and this green sheet are kind of smushed together. And so this is the beta sandwich structure. It's like a flattened beta barrel, if you like. And then in the variable domain, you can see that instead of 7, we now have 8. And the 8th one is really short, but we have 8 little strands that make up our beta sheets. And so we have a bigger beta sandwich, and that's what a light chain looks like. The 2 immunoglobulin-like domains, even though the one has 7 strands and the other has 8 strands, the overall way that they are hooked up to each other is similar, and each of these does have a disulfide bond that you can see here. It's a little black line joining up some of the strands in here. And so we give them different names, but really the light chain domain up here just has 2 extra, little short beta strands as part of that blue beta sheet. I missed one when I was counting earlier. There's 5 of those. And the 4 red ones here. And so the red ones are equivalent to the yellow ones, and the blue ones are equivalent to the green ones, but there's an extra 2 on the end. That's the only difference between the variable and the constant domains of the light chain. And it's going to look similar in the heavy chain. I think we will get to a heavy chain a little bit as part of a whole antibody. All right, so what are some of the other parts of the antibody? Well, one part is the hinge region. And let's see, the hinge region is going to be right here and right here. So this whole arm can rotate. So this part right here can rotate all the way down to here, all the way over to here if it needs to. It's going to be quite flexible. And so that's the hinge region. It's just a thin little bit of protein that is rather exposed and that we can cut into pieces. And so those 2 pieces are going to be the FAB, which is the part that binds the antigen binding fragment, and the FC, which is going to contain only constant domains and is going to be the thing that other cells of the immune system are going to hold on to. And so IgGs and IgAs, for example, have a lot of flexibility in the hinge region. And so that lets them bind to a wide range of things. Okay, papain, P-A-P-A-I-N, is a protease. So it's a protein cutting enzyme. And way back in the day, early immunology, they used to take antibodies and they would put this papain, papaya enzyme is one thing that people call it. It's sometimes sold as a supplement, although by the time it goes through your stomach, it's actually broken down and it's just protein, same as any other protein. So it doesn't have any magical papaya properties. It's just a source of some protein, a very expensive source of protein. It is able to cut proteins and is able to cut in the hinge domain of the IgG antibodies. And so it makes two fragments, and the old timers used to call those, oh, that's the fragment that binds the antigen, so antigen binding fragment, FAB. This one is the constant fragment that seems to be the same for all these IgGs, I guess we'll call them, and so that's the FC. And so yeah, they're just two fragments that you get when you use papain to digest an antibody. And so FAB fragment and FC fragment are exactly what we said, and you can put those on your cards just to make sure. Now there's another way. These old timers didn't just use papain, they also used pepsin. And pepsin cuts antibodies in a neat way. So it'll cut IgG antibodies specifically, which are the most common ones in the blood. And it'll cut them just after the part that links the two sides together. So after that disulfide linkage. And so we call these fragments the FAB prime 2, because it's basically two FABs, and they're a little bit longer, so it's got the little apostrophe there, which we would say FAB prime in biology. So yeah, it's got two linked FABs together with no FC region at all. So it's a thing that can bind to two antigens and cannot be used in any way by the immune system, but is useful to play around with in the lab. You don't see these quite as much anymore, but they did exist. And if you ever run into one, this will tell you the difference. So here's a nice little graphic explaining what's going on here. We've got papain, which cuts right here. You see it's just above these two linkages and just under this linkage. So when you cut, you're going to get two FABs and one FC. And they are both disulfide bonded together, so they're both going to stay together, yeah, forever. But when you cut with pepsin, it makes multiple cuts. And all the cuts are actually below the disulfide linker here. And so you'll make this thing, the FAB prime 2, and you'll kind of shred your FC, but you can call this a proteolytic FC prime is another way to describe it. There's not a lot of use for these things, but if you run into one in your immunological career, that's where it came from, and just so you've seen it once. But mostly we're going to be dealing with papain-cleaved FABs and FCs. And this is actually very easy. You can buy a kit, and it'll do this quite well, quite efficiently. Okay, another little term for you is called a hapten. So a hapten is any molecule that could be recognized by a antibody, but can only be recognized when it is stuck to something else. So sometimes you just have to have two haptens stuck together and then an antibody can recognize them. Or sometimes a hapten actually has to bind onto a protein, for example. And our beautiful example of this is urushiol. This is the compound in poison ivy and poison oak and poison sumac that makes you swell up and go itchy and just, yeah, makes life that much worse. What you get with urushiol is actually an immune response, and it's going to be driven by T cells. The first time you catch poison ivy, it generally doesn't make any immune response that you can see. It doesn't cause that reaction on the skin. But in that first time, you're going to make little T cells that are ready to go out and protect you. And the second time, you're going to make a mighty response. And so you'll swell up and you'll just feel terrible. But this is going to be from the T cell driven inflammatory response. Urushiol is an example of a hapten because you can't actually bind it with a T cell receptor or an antibody unless it has already bound to a protein. And urushiol will bind quite readily to a variety of proteins in your skin. It's just a reactive little compound that once it is bound there, then you make a very strong immune reaction against it. And so that's the story of poison ivy. It's all down to immunology. Now those hinge regions that we were talking about, and the hinges are going to be right here and here and here and here and here, they allow antibodies to join together on these little haptens. So this is a hapten, and there's two hapten molecules that are bound together. That's the two red things. So on their own, they're not big enough to bind to an antibody, but together they are now a legitimate antibody target. And so since these arms can bend and flex all over the place, they can bind to little haptens in different configurations. And it's not just for binding haptens. They can bind to proteins on the surface of a virus, for example, that may be farther apart and require a different angle between the two domains at the top, the two FAB domains. Okay, so that's the end of that section, and then we're going to go in and look a little more deeply at antibody molecules, and then the second half of the chapter is all going to be about T-cell receptors. So I would advise you to break this up into multiple weeks. Let's just hit all the antibody stuff in the first week, and we'll do all the T-cell receptor stuff in the second week. Thanks very much.